haploinsufficiency, gene dosage. Number 3, gain of function. Yeah. Okay, great. So as promised, we're going to go over some pedigrees today. Most of them don't have tricks, so don't look for tricks. I'll tell you the ones that are tricky.
You know, the key word there is some of them.
Yeah. And you'll definitely have a pedigree question on your comprehensive exam. Okay, so we'll start out with a very easy one. Anybody have any thoughts about this pedigree and the inheritance pattern? Well, first of all, let's start at the very beginning. What are the squares?
Males.
What are the females? What are the black or filled symbols?
Affected phenotype.
Right. And what is the open circle? Right. Okay. So here's the pedigree. The parents are unaffected, and there are multiple affected children, both male and female.
Autosomal recessive.
Autosomal recessive is the most likely inheritance pattern. Right. Very good. And this is an example of the kind of analysis that was done before high-throughput sequencing was available, where you have to find a restriction enzyme whose site overlapped the site of the change, so that you would have a gain or loss of the restriction site. And so here you can see that you can see the parents here. So there are two different missense alleles that are being inherited from the mother and the father, okay? And you can see one changes which amino acid? Arginine to glutamine at 229. The other changes arginine at 291 to tryptophan, okay? And you can see that this is the sort of the diagnostic restriction fragment. You see this middle band, and then these two bands on either side of it for this one. And you can see which band is diagnostic of the mutant allele? The top band here. Yeah, you see it's present in all of the affected individuals. And then down here, which is the diagnostic fragment? The bottom one, yeah. You can see that the father has the top one, but not the bottom one. The mother has the bottom one, but not the top one, and the affected kids have both. Okay? So that kind of illustrates the idea that oftentimes with the recessive disorders, there will be two different mutant alleles. When do you always have the same mutant allele? From being inherited from both parents. Under what condition? All right, well, hold that thought. We'll get there. Okay, and then this is just highlighting the idea that these are missense mutations, and how do we know that this change in the amino acid has had a significant effect on the function of the protein? Well, in this case, one of the arginine residues you can see is conserved in all of the species shown on the slide, all the way from humans to flies. So that implies that it's pretty important to have arginine at that site in the protein, okay? And other residues are not tolerated. So that gives you a clue that that missense really does have functional importance. And there are now algorithms so if you find a variant in a patient and you want to try to figure out, is this something that is a functional variant, or is it just a polymorphism? There are programs that give the likelihood of it being a pathogenic allele. Okay, so we have those tools now. All right, here's another pedigree, and in this case you can see again, there are multiple affected offspring in the second generation. And this is a disease called hereditary erythrocytosis, where the affected individuals make too many red blood cells. So you can see the hematocrit, which is normally at 45, the mean hematocrit is 70 in these patients, and some of them have much higher than that. And you can see that this appears to be due to an increase in the production of erythropoietin, the hormone that controls red blood cell production, because the patients also have elevated EPO levels. So what's the inheritance pattern here? Again, multiple affected in the parents, unaffected, males and females.
Looks again like recessive.
Yeah, autosomal recessive. Autosomal recessive inheritance. Okay, and it turns out that in this disease, in this particular population, there's a single mutant allele that segregates this R200W missense mutation, and in this gene called VHL. This is in an area of the former Soviet Union called Chuvashia, and I guess it was somewhat isolated, and as a result, there's this allele is present at a very high frequency in the population. And what factors would lead to a high increased frequency for a deleterious allele in a population?
Increased inbreeding, usually a small population size.
Right, so if you have a small population size and then just genetic drift where the frequency of the allele is high, and you don't have new alleles being put into the population. And then what's another mechanism for a deleterious allele having a high frequency in a population?
Or like the same reason bottlenecking.
Okay, so I'm thinking about something that, a reason why a mutant allele would be selected. An allele that's deleterious in the homozygous state would be selected. What's an example of that?
So, I mean, it could be if heterozygosity confers some survival benefit, like in the sickle cell, disease where
Right.
having heterozygous, protects you from malaria.
And why is that such a powerful selective effect compared to the negative selective effect in the homozygous?
Is your question why is it like overcome the negative effects of the homozygous? I guess it just depends on how powerful that selective pressure is, and also if it's just still unlikely enough that it's homozygous. Not sure.
Right, so are there more heterozygotes or homozygotes in the population?
Heterozygotes.
Many more heterozygotes, right? So because there are many more heterozygotes, the effect of the selective force is greater. And that's how you end up with a high frequency of alleles like sickle cell and thalassemia, because they protect against malaria. In this case, it's not known whether this is just due to genetic drift or whether it's due to a positive selection in, say, the heterozygote state, or even could be in the homozygote state in some particular, in response to some disease, it may be protective, right? So it's not really known here. Okay. All right, so here's two more pedigrees. And what's different about these pedigrees?
They're cosanguineous.
Say again?
They're cosanguineous.
And how did you know that?
The double line.
Right, so these double horizontal lines refer to consanguinity, and what is consanguinity?
Normally, I think it's first cousin or closer, but general close
Right,
relationship.
so individuals who are related and have a common ancestor. And why is a common ancestor important? Thinking about rare genetic disorders.
Well, you're implying, if you have a common ancestor, you are, at least some of your two gene pools are pulling from the same pool. So if your common ancestor had some rare recessive disease, or rare recessive allele, even one allele, you both
Only
could
takes
have
one,
it.
right? That's the point. So it only takes one rare recessive allele because that can be inherited on both sides because of the common ancestry. So the rule is the rarer the disease, the more likely it is that an affected individual will be the result of consanguinity. Okay? Because as the disease becomes rarer, the likelihood of two unrelated people carrying the mutant allele coming together is very low. But it's very high when there's identity by descent, meaning the same exact allele has come down to both partners. So here we see that, and so that means the inheritance pattern is what here? Autosomal recessive, yeah. Okay, and again, these analyses of the alleles show that, and they show again that the alleles coming from the mother and the father are the same. They have the same pattern, right? And the affected children, of course, have only that pattern because they're homozygous for that allele. Any questions about that? So that's one of those things that's very helpful to keep in mind. All right, here are two pedigrees for a disease called familial hypercholesterolemia. And in this disease, there are elevated blood cholesterol levels, and those are indicated numerically on the pedigrees. And then you see these filled and half-filled symbols. So what did you make of that? And so who are the affected individuals? What are the symbols? Yes, you can see these are the ones where the cholesterol levels are elevated. And what about the half-filled symbols? What do you think those represent? Carriers, yeah. So that's possible. And then if that were true, what would be the inheritance pattern? Autosomal recessive, right? So you say, well, why were these published? It's a known disease. Well, it turns out this known disease is autosomal dominant, inheritance in most cases. So here we have autosomal recessive inheritance, and why do you think that would be? If the disease is normally autosomal dominant, but here in these two families it's autosomal recessive. What would be potential explanations for the different inheritance pattern in these families compared to most?
I know there's different types of familial hypercholesterolemia with different inheritance patterns, but I don't know if that's what you're...
Right, so at the molecular level, what could be an explanation?
It's a different SOD.
So it's a different mutation that has different properties, like the types that we were talking about. Okay, so that's one explanation. What's another?
It could be that there's varying efficacy of the other allele that they happen to inherit. So if it's a dosage problem, if they have this other allele that happens to be strong enough to overcome that dosage.
Right, this is autosomal recessive, right? So here the affected individuals are affected to mutant alleles. So one possibility, so we talk about different types of mutations in the same gene. Of course, they could be different mutations in a different gene, right? And in order to inactivate that gene, it requires both copies being inactivated. And that's the case here, where these are patients who, rather than having a mutation in the LDL receptor gene, which is usually the case in this disease, they were found to have a mutation in this other gene. So Yes, so you have to be careful that you can't be too dogmatic about a particular disease having a particular inheritance pattern necessarily. But these are exceptions, that's why they get published. Okay, so then you can have mutations either at the LDL-RA Arch1 gene giving rise to familial hypercholesterolemia. So what's that called? Mutations at two different loci? Genetic heterogeneity, yeah. That's the one. Okay, so this just summarizes the features of autosomal recessive inheritance, that you have this so-called horizontal pattern where you have multiple affected in a single generation and the parents are unaffected. So again, if these two individuals wanted to have another child, the risk of recurrence in that child would be 1 in 4. And as I mentioned, the frequency of consanguinity among the parents is inversely related to the allele frequency. Okay, here's another pedigree. This is another pedigree from another family with hereditary erythrocytosis. So remember the one I showed you previously was autosomal recessive inheritance. And here we have a 4th-generation pedigree with affected individuals in 3 generations, both male and female, with at least 2 affected parents giving rise to affected offspring. So what's the most likely inheritance pattern here? There's no trace. Autosomal dominant, yeah. So here, rather than that horizontal pattern, now we see a vertical pattern, right? Because again, just one mutant allele is sufficient to give rise to the phenotype. Okay, and so this turns out to be in a different gene. We said the first type was the VHL gene. This is in the EPAR receptor gene. So now we have 2 different loci that can cause hereditary erythrocytosis. So what's that called? Genetic heterogeneity, just like it was 2 minutes ago, yeah? Okay. Okay, so here again, in the olden days, we would find variants at a locus, and to see if that locus might be the site of the mutation, we look at the segregation of these variants in the individuals in the pedigree. So the variants are shown here, they're like 1 through 11. And the question is, which variant is on the allele that's disease-causing in this family? So here you can see 7 and 8, 8 and 8, 7 and 8, 6 and 8, 8 and 11. Eight is the one that all of the affecteds have in common, right? Okay, and then what about the first generation? Do you think that either one of the parents was affected in the first generation? Could they be? Could you rule it out?
Father might have been, but
Right, because he's got the 8 allele, right? So he may have been affected. And if he was 50 when his child was born, what would you also think about? A de novo mutation, yeah, starting the disease incidence in the family. Yep. All right, so advanced paternal age, new dominant mutations, right?
So for that one,
Mhm.
the fact that some of the siblings also have the 8 And that's, I assume that's a restriction like fragment polymorphism, right?
Right.
So the fact that they have it suggests that it wasn't necessarily the father. Yes? No?
You mean in this generation?
Yeah.
See, in this generation, so in this generation where you have this individual who have that, who inherited that allele, yeah, it sure looks like it's a new mutation. It would be nicer if this individual was this individual, right? Because the father would be older when she was born. But anyway, yeah. So I agree that would argue in favor of it being a de novo mutation. But unfortunately, the father's not available, so we can't confirm that.
Could you argue that 11 is protected, or could be protected if it's in 4?
You mean here and here?
And, yeah.
It's possible, sure.
But you'd also have to argue 6 is protected since the other sisters is 6/8.
Yeah, so that's not as clean, yeah. Okay, great. How about this one? So here we have, again, 4 generations of affected individuals. And now, in this family, it's reported that in addition to elevated red blood cells, they also have pulmonary hypertension, where the blood flow to the lungs is at increased pressure, and this is a serious medical condition as well. So, and it's present. You can see 2 of the individuals in the 2nd generation. So, all right. So now we have erythrocytosis and pulmonary hypertension. What does that make this?
Pleiotropic?
Right, but what does it make it as a disease entity? A syndrome, right? Now we have more than one phenotypic manifestation. We can call it a syndrome. Yeah. And what's the inheritance pattern?
Erythrocytosis is still autosomal dominant.
Yeah. And yeah, remember for the dominant disorders that you can have variable expressivity, right? Where you may have multiple phenotypic manifestations. Some affected individuals have some of them, others have others. Obviously, if the name of the disease is erythrocytosis, everybody has to have that, right? Because that's the means of ascertainment. And yeah, I mean, everybody with the disease gets that. What could you argue about pulmonary hypertension in terms of the pathogenesis? Why it's only present in the 2 individuals in the 2nd generation?
Just age, maybe?
Yeah, so in other words, it may be age dependent. So that can be a tricky aspect of genetic disorders, where someone, non-penetrant is simply that they haven't developed the manifestations yet because they are age dependent. Okay, great. So this is now the EPAS1 gene. So now we have another gene involved. All right. So here's another pedigree, and what this pedigree is showing that is there are individuals who have erythrocytosis. It's due to a mutation in this EPAS1 gene, but these individuals also have, some of these individuals also have thrombotic complications, where they'll have a myocardial infarction or a stroke or a pulmonary embolism. So this is yet, in this family, yet another part of the phenotype. And you can see for different individuals, they have different manifestations, right? So again, the bottom left is showing that they have inherited the mutant allele, okay? and they've inherited one copy of the mutant allele. So everybody who has this, some of them also have this. So what's the inheritance pattern here? So just looking at erythrocytosis, you can see it starts with people in the second generation, third, every generation.
Still autosomal dominant?
Yeah, still autosomal dominant. And again, we have this additional phenotypic manifestation in some of the affected individuals and not in others. What do we make of this individual?
He could be lucky, or it could be non-paternity for his children.
Okay, so I'm asking about the thrombotic complication. What does it do to in that individual? Some other cause, right? So that's very common, a set of diseases, right? So that's another thing that can is a potential obstacle to understanding pedigrees. If someone has similar phenotypic manifestations but due to another cause. And that's likely in that individual. And yeah. Okay, great. So this is autosomal dominant inheritance. And it's due to just a single missense mutant allele. And we can now call this a syndrome. Okay, so you can have mutations in the EPAR receptor, VHL, or EPAS1 gene that cause hereditary erythrocytosis. So that's what? Genetic heterogeneity. Yeah. All right, here's another pedigree. This is a disease called familial partial lipodystrophy, which involves handling of lipids, metabolism of lipids in the body. What's the inheritance pattern here? Yeah, it's autosomal dominant, again, affected, the offspring of affected parents, both sex affected. And then there's a variety of phenotypic manifestations. And you can see that for some of these, like diabetes and hypertension, some affected individuals have those manifestations, others do not. So again, there's variable expressivity with regards to different aspects of the phenotypic presentation of the syndrome. And as I said, it's very common in autosomal dominant disorders because you have the wild type allele. So it gives you a much more variable outcome. All right, and this is variable expressivity. Okay, so here's another pedigree. Oh dear, this is that syndrome. Besides the individuals with the filled circles, is there anybody else in the pedigree who must be carrying the mutant allele? Is there an obligate carrier here? And if so, who would that be? 1, 1, 2, 1, or 2, 2? 2, 2, yeah. 2, 2 has to carry the mutant allele to give it to his offspring. And he received it from 1, 2. Okay, so, and what's that called? What do we say about this individual? Non-penetrant, right, for the phenotype, yeah. So the pedigree shows incomplete penetrance. Sorry for making you read my mind. I apologize. All right. So here's an interesting disease. This is called the Waardenburg syndrome. And the affected individuals are heterozygous for mutation at a gene called PAX3. You remember PAX6 was the cause of aniridia. So what's going on with the eyes of this woman? There's two things. One, you can see that these vertical lines were drawn so that they could measure this distance, and that's called the intercanthal distance, and that's increased in this individual and individuals with this Waardenburg syndrome. What's the other striking aspect of the eyes? No, sorry, it's a black and white photo, but they're very pale, right? The irises are a very pale color. So we have words for that, hypoisochromia iridis, right? Where there's decreased color in the irises and it's equal in both eyes. Why is it important to stress that? Well, because there are individuals who look like this. And what's up with this guy? Two different colored irises, right? So we call that heterochromia iridis. He's got different colored irises. So this is an audit. So this highlights the fact that with autosomal dominant disorders, not only will you have variable expressivity with regards to different individuals in the pedigree, but even in a single individual, there may be variable expressivity of paired structures in the body. So how do we count for that? Let's say, this individual, his parent is affected, right? And he has multiple affected siblings. So how do we count for the fact that one iris is brown and the other is a pale blue, with pale blue being the affected iris, if you will? What do you think determines blue or brown? Should we flip a coin? Yeah, we should flip a coin. Because remember, I told you about stochastic events, right? And probabilities. So here's the probability. And in one case, it came up heads, and the other case, it came up tails. Now, this coin does not have just two sides, right? It has many sides, and there are many more tails than heads. Okay? So the likelihood is you'll end up like this. But there is a discrete likelihood that you'll end up like that. And as I said, this is very common in autosomal dominant disorders. Very common. Okay. What do we have going on here? Yeah. So what's the first thing the obstetrician does? He counts fingers and toes on the baby. Sometimes, unfortunately, he does that despite the fact that the baby's not breathing and needs to be given to the pediatrician, but that's a different story. What do we have here? We have 4 fingers. Count them. What do we call that? Oligodactyly, a very nice Latin word, meaning few fingers. What about on this hand? What's the abnormality? There is no abnormality. It's completely normal, right? So one completely normal hand, one hand with only 4 fingers. This individual has a mutation in a gene called TBX3 and has what's called the ulnar mammary syndrome, which affects the ulnar side of the arm, okay, meaning pinky and the ulna. And then in females also affects mammary development. Autosomal dominant. Okay, so here's our summary for the autosomal dominant inheritance. Again, this vertical pattern with affected individuals who have affected parents and offspring. There can be a new mutation, right, that starts the disease transmission. And in that case, paternal age effect may play a role. The risk of recurrence is 50%, right, because the affected parent has one affected, one unaffected allele, and the chance of passing them on is equal. And so when we see a parent where the parents are unaffected, they have an affected child, and it's a known autosomal dominant disorder, then what do we counsel them, or let's put it this way, what are the likely causes of the fact that the unaffected parents gave rise to an affected child for a disease that's autosomal dominant inheritance pattern? What are the explanations? There are several.
Non-paternity.
It could be non-paternity, thank you very much. Yes. What else could it be?
De novo mutation.
It could be a de novo mutation in who? Blame the father, yeah. The mothers always feel guilty, so blame the father. No, I'm kidding. It could be in either one, but again, we have the paternal age effect, right, that may tip you in one way or the other, but either one could certainly be. And then what's the third possibility? It's a much rarer possibility.
Father has a germline mutation.
The father has a germline, or it could be the mother, right? So, in other words, and is that in the father or the mother who have a germline mutant allele, is it present in every cell of their body? No. And what do we call that?
Mosaicism.
Yeah, mosaicism. So, in other words, the mutation arose during the somatic development of the parent, right? And it arose in the lineage that gave rise to the germ cells and was not present in the lineages that give rise to the phenotype, right? So there's, and if it, of course, if it affects all the germ cells, then what's the risk of recurrence to the couple? 100%.
Well, if
It's autosomal dominant disorder.
Are you asking about the offspring?
Yeah, what's the risk of recurrence in that case, where you have germinal mosaicism giving rise to an affected child? 50%, right? Because it only affects one allele. Yeah. Okay. So, and then of course, if it occurs very late and only affects some of the germ cells, right? Then it's going to be even lower potentially. So some dominant diseases, there is a kind of recognizable level of germinal mosaicism. And so in those diseases, the recurrence risk for parents who are unaffected and have an affected child might be like 1 to 3%, okay, just based on the numbers. And what kind of genes do you think either new mutations or germinal mosaicism are more likely to occur in? Big genes or little genes?
Big genes?
Big genes, right? It's a bigger target, right? So in general, big genes have higher mutation rates than little genes, everything else being equal, right? Yeah. Okay, and so like Duchenne muscular dystrophy, we talked about that big gene. Yeah, there may be a recurrence risk there. That's on the order of 1 to 3%. Okay, so here's another disorder. This one affects the eye, resulting in vision loss. And what's the most complete description of this pedigree from a genetic point of view? X-linked. X-linked what? X-linked recessive, yeah. So the mutation is on which chromosome? The X chromosome, all right? The male is hemizygous, so the mutation is uncovered. He transmits the disease to his daughter, the mutant allele to his daughter, who also inherits a wild type allele from her mother. So she's protected, she's heterozygous, she's a carrier, and then there's a what chance that she'll pass it on to her son? 50% chance, right? Because she's got two X chromosomes. One has the mutant allele. And if the male offspring receives that chromosome, he will be affected. Yeah, so that's your typical autosomal recessive inheritance. And when new mutations occur, so for example, if this father appeared to be unaffected and there was an affected male, who might be the source of the new mutation? Especially if that particular parent is older.
So it could be that father, that grandfather.
The grandfather, right? Grandpaternal effect in X-linked recessive, right? Compared to paternal effect in autosomal dominant, because it takes the 2 generations to uncover the mutant allele. Okay, and yeah, this is just showing one affected, one unaffected allele. So here's the scoop on the autosomal recessives. So not only are most of the affected individuals male, but there cannot be male-to-male transmission. So let's think about exceptions to that rule. When might you have an affected female? Or what circumstances might give rise to an affected female?
If X inactivation, so if you have the healthy allele that is inactivated, you might.
Right, so if the wild type allele, if they're skewed X inactivation, right, normally it would be random. You would get 50% of one X being inactivated in some cells and 50% in other cells, the other X is inactivated. But if by chance or some selective force that skewed toward inactivation of the wild type allele, then the female would be like the male, right? In terms of the only active X being the mutant one.
Couldn't like Turner syndrome also cause it? If you only have one X chromosome?
Yes, if you only have one X chromosome, as in Turner syndrome, that would cause it. And what would be a case where you might get male-to-male transmission?
Klinefelter?
Yeah, if the father, right, had Klinefelter, no, if the father would have to have the Klinefelter syndrome, right, and would pass an X and a Y to the son, and then if he got an X from the mother,
But it doesn't have some sort of protective support.
right, but then he would have to have like somatic non-disjunction and lose the wild type X in some of his cells, yeah. So not too likely. Okay. Here's the pedigree where you see there are affected individuals. In the first pedigree, it's in just one generation, but in the second pedigree, it's in 2 generations. And they're all females that are affected. And then there are females with little bullseyes. So this is like somewhat of a trick pedigree, I guess you would say. So what inheritance pattern do you know that would result in just affected females? Where would the mutation lie?
I'm not confident, but like mitochondrial disorders.
Say again?
Like at least mitochondrial disorders for the offspring, but
So yeah, in the mitochondrial, the mother is the source of the mutant mitochondria. Here, though, we're talking about the affecteds being females only. So yeah, what would account for the only females being affected?
Well, as far as I can tell, there is a male who has a homozygote.
Yeah. So this, like I said, these pedigrees are a bit complicated. That's why they get published. Okay. So what are you thinking if there's this male? There is a male who's affected, but
So it could be X-linked dominant, where
Mhm.
the females who get it are affected.
right
The reason for males to not be affected, though,
is what?
maybe a dosage deal, but I don't know.
It could be that the males who are affected just aren't born.
Exactly. Yeah. So the males, obviously there are males there again, but they're hemizygous, right? So the disease occurs even in the females who have a wild type allele. So in the male, where there's no balancing wild type allele, it's much more severe, and the pregnancies are lost prior to birth. So you don't see affected males. You also see a reduction in the, if you have a large pedigree, in the number of male offspring, right? In general, you don't see any affected males, but in this rare case, somehow there was an affected male. And then the other thing that's unusual here is there are unaffected female carriers, right, in both of the pedigrees. That's what this means, that those females have the mutant allele but didn't show the phenotype. There's a lot of unusual findings here. This is one of Kirby's favorite diseases, Rett syndrome. Any comments you want to make about that, Kirby?
No.
This pedigree is the lighter shading for the male is that anything, or is that just shows that?
Yeah, I don't recall exactly how the phenotype was different in the male compared to the females, but you can imagine that it would be different. And I think that's what the different shading is trying to denote. Okay, so again, here we have this restriction pattern that shows the two alleles, right? The wild type allele is this 351 band, and then the two smaller fragments are the mutant allele. And you can see that's the molecular proof, I guess, that the female is a carrier. And likewise here, where it's the upper band, it's a different analysis. Probably it means it's a different allele in this family compared to that family. And you can see that this upper band is the mutant allele, so this male has it, right? Doesn't have any trace down here. Now, of course, this is probably from blood, right? The DNA analysis is probably blood cells that are being analyzed. So thinking about this, what might be a mechanism by which that this affected male might be surviving? Is there some mechanism like of mosaicism that would allow him to survive? I don't see any unless he was XXY, right?
But then both his X's would have to be affected, because we don't see the
Exactly. Well, again, in the blood, right? In the
blood.
blood, yeah. But yeah, it would be, require some complicated gymnastics to, to get him to that outcome. Yeah. So it may be, for example, some modifying allele at a different locus that is enabled him to survive. Okay, and here is the rundown, in general, you only have affected females. And there's a 50% recurrence risk, but only in the daughters of affected females. Okay, how about this pedigree? So in this pedigree, these years refer to, up here, these are the age of the individuals at the time the pedigree was taken. And these numbers here refer to the length of time that the individuals were been trying to have an offspring. Okay, so they're infertile. Here, 4 years without an offspring. Here, 5 years before an offspring. Here, 5 years, she gave up, tried him, that worked out much better. And here, 3 years and zip. So, what do you make of that? Yeah, Y-linked inheritance. And what's a requirement for Y-linked inheritance? What kind of transmission? You have to have male-to-male transmission, right? Yeah. So, this is Y-linked. Only males affected, and there's male-to-male transmission. And what do most of the genes on the Y chromosome do? Make sperm, yeah. So the phenotype for Y-linked mutations is often infertility. So what do we make of this one up here? 16 years. This individual turned out not to have the mutation present in all the other infertile males. There's lots of genes on the Y chromosome. So that individual presumably has infertility due to some other cause. So again, that makes the pedigree analysis. So the affected individuals with the black symbols all had a microdeletion, whereas this guy had lack of sperm but no detectable microdeletion. So presumably has infertility due to some other cause.
So was this 2-1, the grandfather, did he just happen to be lucky and still, be able to have 5 children?
Yeah,
Is there
right.
just some lining up with his wife that made their fetuses more viable? What do you think?
Yeah. Again, usually what it is, it's a matter of production of the sperm, right? So this shows and obviously there's lots of variability in terms of some individuals being able to have offspring and others not. So it suggests that there might be other modifier loci that are affecting the phenotype.
And also, does it seem significant the fact that he pretty much only had sons, with the exception of just the one daughter?
Well, that's the luck of the draw.
Yeah.
Because, I can't imagine a mechanism that would also affect the sex selection. Although, again, as we know, it's dependent on the sperm, not the egg. So, that was Henry VIII's mistake. Okay. All right. right, so this pedigree now is an interesting one. It's got a little something extra going on. So to interpret this, the affected individuals, again, are the filled symbols. This is the restriction enzyme digest of the locus in question. Okay, so you can see what the genotype of the individuals are, who's carrying which is the mutant allele. 200 or 175? The 200, yeah. And you can see that there are some individuals who have the mutant allele but don't have the phenotype. They're denoted by the dot, okay? And you can see there are both males and females who are carrying the mutant allele who don't have the phenotype, just like there are males and females who do have the phenotype. And you can see there is parent to offspring transmission. So if you focus on the cases so here you have an affected male and he has 2 affected offspring and 1 unaffected offspring, and here is a male, right? And he gave rise to a female who's carrying the mutant allele and has the disease, right? Whereas this female has the mutant allele, passes it on to her son, but he doesn't have the disease. And you can see that this is an affected female, and she had 2 sons who are carrying the mutant allele but are not affected. And her sister, who's affected, gave rise to a son who carries the mutant allele but is not affected. So the only way that you get the disease, your father carries the mutant allele, right? And you don't get the disease if the mutant allele is passed on from your mother. So what's going on here then? What's the word we use to describe this phenomenon?
Imprinting.
Imprinting, yes. So this is imprinting, and what's the general concept of imprinting? What's different between the allele that comes from the mother and the father at an imprinted locus? Right, well, so only one of the alleles is active, okay? So unlike most loci, right, where you get a copy of the gene from the mother and the father and both are active, in the case of imprinting, one of the parental genes is inactive, right? So when it goes through that germline, it's inactivated. And the idea is if an individual has a wild type and a mutant allele, but the wild type allele has been inactivated, then there's no functional copy of the gene, right? And the individual has the phenotype. Whereas if it's the mutant allele that's inactivated, well, who cares, right? It's already inactive. You have the wild type copy and you don't have the phenotype. Okay, so, and this is an unusual one because this is actually tumor predisposition syndrome, where the gene appears to be imprinted in the relevant tissue, which is some sympathetic nervous system, part of the sympathetic nervous system. Okay, so this is imprinting. So you want to look at what happens when the allele is inherited from a male versus a female, okay? And that's the key to imprinting. So you might say, well, it looks like autosomal dominant with reduced penetrance. It is that way, right? But it's a kind of a special case because it has to do with which allele comes from which parent. Okay. So this is a much more common imprinting disorder called Angelman syndrome. It's a syndrome characterized by developmental delay and other neurological findings. And in this disease, the imprinting involves a gene called UBE3A. And you can see in this pedigree that when the mutation is passed from the father to his children, they are unaffected, but when a female transmits the mutant allele, her children are affected, okay? So this means that, so which allele is inactivated in is it the paternal allele or the maternal allele? The paternal, right? Because the mutation is on the maternal allele, and if the paternal allele is inactivated, then you have no functioning copy of the gene.
Are there any therapies that are basically seeking to bring up the expression of?
Right, so as was mentioned, the imprinting often involves methylation of a gene, and obviously there are DNA methyltransferases that mediate that, and some inhibitors of those are used as potential cancer treatment. Kirby, do you know if they've been tried in imprinting disorders?
I don't know.
I don't think so. Yeah. But it's a good idea. Okay, so the question is, in some of these patients, there's actually a chromosomal deletion. So this area of chromosome 15 is deleted. That's the basis of the mutation. Okay, and the deletion encompasses this UB3A locus. But of course, you could also have a loss of function mutation at that locus that does not involve a deletion. And those have been found as well. And but again, the same inheritance pattern holds. And then the question is, could you have Angelman syndrome without any mutation at all? Question?
I just had a thought about it. Is it possible that you have uniparental inheritance, so you just get both alleles that you have are silenced paternally?
Yeah, that's right. So, and how would that arise? How would you end up with two, let's say, paternal copies and no maternal copy of chromosome 15?
Some meiotic recombination event?
So if you had meiotic recombination and one of the germ cells had two 15s, the other had one, then you would have a child with what? Trisomy 15, right? And is trisomy 15 viable? No. So how would you get from trisomy 15 to the isodisomy?
If you had non-disjunction in both, or if you lose the chromosome 15.
Right, so if you have somatic non-disjunction, right, during mitosis, So you lose one of the 15s. Now you're back down to 2, which is the right number, but they're both from the same parent, right? And that doesn't work. Yeah, so that's thought to be the mechanism for isodisomy, is that first you have meiotic non-disjunction adding a 15, then you have mitotic non-disjunction subtracting a 15 in some subset, right, depending where it occurs during development, presumably in early development. Okay, so these individuals are likely to be mosaic. All right. So, yeah, in this case, paternal uniparental disomy. All right. Okay, so the other interesting thing about this is that in the same interval on chromosome 15, again, this is a fairly large interval, there's another disorder called Prader-Willi syndrome where there's an imprinted allele, but it's the opposite in terms of which allele is active, okay? So it involves a different gene, and it's the opposite pattern of imprinting, and it's got a completely different phenotype. But it was very interesting because initially, when the diseases were just studied at the level of the karyotype, there were deletions that looked the same in these two different disorders, right? People are scratching their heads saying, how do you get? So, but it turned out that the critical interval in the deletion is not the same. It's a different gene target. But in this same region, but in this case, the deletion is always on the paternal allele, whereas in Angelman syndrome, the deletion is always on the maternal allele. And there have been, again, cases of uniparental isodisomy giving rise to Prader-Willi syndrome as well. Okay, here's a really large pedigree, lots of offspring, and it's for a very terrible disease that causes both blindness and deafness. And you can see that there are multiple affected individuals in multiple generations, and both males and females are affected. And again, look at the parent of origin. So for affected individuals, where there's an affected parent, who did the affected individual, what sex is the affected parent? Female. So in other words, you always have an affected female when there's affected children. So what's a likely mechanism here? Could it be imprinting? So
Mitochondrial.
yeah, so it could be mitochondrial because the mitochondria, of course, all come from in the egg, right? So if a male has it, he doesn't transmit it because there are no mitochondria in the sperm. And so you get this situation where you can have virtually all of the offspring affected, right? As opposed to like the imprinting where it still is only going to give you 50% affected, right? But here you can have up to 100% affected. Again, depending on what kind of distribution of, affected versus unaffected mitochondria there are, and how they get distributed to the offspring through the egg. So that's those are the highlights of the mitochondrial inheritance.
Question about this pedigree. On row 5, I don't think they were given, I can't see what the number is, but on the far left, the affected female,
Yes?
curious about her as far as like
Yeah.
mother nor grandmother, if that's a great grandmother.
Yeah. Yeah, so that's a really interesting one, right?
Idiopathic or something.
Yeah, so the question is, so let's look at the inheritance. So there was an affected female, and then an unaffected female, an unaffected female. So it is coming through the females, right? So it certainly could be, right, the same mutant mitochondria, right?
Just in low percentage.
Yeah, which were, again, the two of the females got a high proportion of wild-type mitochondria, but some mutant mitochondria. And then this affected female, just by chance, got a very high percentage of mutant mitochondria. That's certainly possible. The other possibility is it could be a new mutation in the egg that went to form this affected female. And it's interesting that, that female has 3 unaffected siblings also, right? So it seems like, this idea of heteroplasmy where you have different proportions of wild type and mutant mitochondria, both in terms of the germ cells with transmission, but then also during life, within different organs, there can be a shift toward a higher percentage of mutant mitochondria. So one mechanism is if the mitochondria have a deletion, then those smaller mitochondrial genomes will replicate faster. And with time, may take over, and that accounts for the fact that these are kind of progressive degenerative disorders that get worse over time. This one is not due to a deletion, however. This one is due to a single nucleotide substitution in a mitochondrial gene. So that's interesting. So understanding here how you would get different proportions of wild type and mutant mitochondria other than just by chance is harder to understand. But it certainly could be just due to chance.
Does the progressive nature of the disorder impact when these patients were screened? So like if these 3 unaffected females were screened earlier in life,
Yes.
it just happened?
Yes. Yeah, for sure they're the youngest. They're the youngest ones in the pedigree. And yes, that's a very good point that that those individuals are still at risk, as are the other unaffected, the individuals who are scored as unaffected in other cases where their mother was affected, right? They're still at risk. Yeah, for sure. Okay. So, so again, the mutation is always in the mitochondrial DNA by definition, but it can be either a point mutation or a deletion. And so you only have maternal transmission. And again, you get this very variable phenotype, and it's degenerative, progressive disorders. And you can see for this disease, which is another disease caused by a mitochondrial mutation, there are deletions, and the percentages of the mutant mitochondrial genomes determine the phenotype. So it takes over 85% mutant DNA for the phenotype to become manifest. And then with over 95%, there's severe disease with early onset. Okay? So, so this is a disease where, again, you can see this evolution, and you can imagine that that the percentage of the mutant mitochondrial DNA might increase over the generations, right? And so you may get more severe disease or earlier onset disease in later generations. And that's referred to as anticipation. And it's seen in one other type of genetic disorder. Do you know what that is?
Like the actual disorder, or?
Just the group category. You can give me an actual.
Oh, I was thinking like Huntington.
Yeah, so Huntington's disease was an example of a trinucleotide repeat. So you have in Huntington's disease, the HD gene has CAG repeated, okay? And in normal individuals, that's repeated 20 to 30 times. And that's in the coding sequence of the gene, so it codes for glutamine, okay? So 20 to 30, affected individuals all have more than 35 repeats. And there's a very interesting paper, I think, in this issue of Cell, where they looked at repeats in brain biopsies from patients with Huntington's disease, looking at a specific class of neurons that are lost in this disease. And what they saw was that, you know, from like 36 to 150 repeats, that occurred slowly over time, like decades. And then like going from 150 to 250, that happened within years. And then going from 250 to up, that happened like in months. And I think it was like over 250 was when the neurons started disappearing. So it's a really amazing study. And what's interesting, of course, is that these neurons are postmitotic, right? So you would think that, because when you have repeats, what do you think of in terms of the generation of the expansion? It's like unequal crossing over, right? If they don't register properly, then you'll get a crossover and you'll get a much larger number of repeats on one of the alleles, right? Smaller number on the other. But these neurons are postmitotic, so it appears to be an error that is introduced during transcription of the gene. So it turns out that during transcription, not only is the DNA being transcribed into RNA, but the polymerase is looking for errors in the DNA as well. And those errors can be corrected, but in this case, it's not an error being corrected, it's a repeat being expanded. Yeah. So the trinucleotide repeats are very interesting diseases. They're autosomal dominant inheritance, right? Because you just have, for Huntington's disease, it's autosomal dominant inheritance. I mean, it can occur on genes on the X chromosome, right? And then it's X-linked inheritance. An example of that is what?
Fragile X syndrome?
Fragile X syndrome, right? Yeah. So, those are, but they're autosomal dominant inheritance, right? But there's this, the changing nature of the alleles, both over time in pedigrees and within the tissue of an individual over their life, that accounts for the fact that the disease onset, so the disease onset is not until the 40s or 50s. You can appreciate by then affected individuals may already have had children. And you can understand the benefit then of having pre-symptomatic diagnosis, okay, which was first done in a study here at Hopkins. That you could diagnose, that you could inform affected individuals that they carried the mutant allele before they showed any signs, or alternatively, that they did not carry the mutant allele and were not at risk for having the disease themselves, and also were not at risk for transmitting the disease to offspring. Okay. I think that's it. All right, so in the genetics clinic, we get a lot of pedigrees that look like this. Oh, I wouldn't say that. We get a lot of pedigrees that look like this, right? And that's usually all we get. But here we have pedigrees where we also have a third generation, and all the pedigrees look like this, where there are affected individuals, but neither their parents nor their siblings are affected. Sorry, well, neither the parents or the offspring are affected. So how would you account for that?
Perhaps a somatic mutation early in development.
So it could be a somatic mutation early in development, yeah. And, okay, but let's think about that. So if that were the case, if it's early in development, presumably you would have cases where the germ cells were part of that, right? And then you should think you would transmit the disease. Why do you think what's the reason why the disease might not be transmitted in that case?
Maybe the germ cells die off or
Right, so it could either be the germ cells that carry the mutation, right, or they can't give rise to a zygote, or the fertilized egg, right, that carries the mutation cannot develop, right? So if it is transmitted through the germline,
it's not viable.
yeah, it doesn't result in a viable pregnancy. So that's the explanation for pedigrees like this, and there are a number of diseases where this has been demonstrated that that's the case. That the mutations are post-zygotic mutations, and that the presence of the mutant allele basically in the zygote is not tolerated. Okay. Say again?
Could that not also have just been autosomal recessive?
No, so if it's autosomal recessive, then you expect that you'll have pedigrees with other affected individuals in the same generation.
But I mean, like, I guess if you were in the clinic and they just had one child, like in the whole.
Oh, for sure. That's the way that
Very unimportant.
that Kirby will torture you in your prelims. Because you're only going to have this, and you're going to have to list every possible mechanism to get that pedigree, right? And there's a lot. Yeah. So good luck with that.
I actually got that question from you, but.
Okay. Anyway, so this is another genetic disorder. What's the inheritance pattern here? Vertical pattern. Autosomal dominant, and are there any non-penetrant individuals in the pedigree? Pedigrees? We have pedigrees A, B, and C, generations 1 through 4.
B2.
B2, 1. Yes. So B2-1, this this female is an obligate carrier because she has an affected parent and an affected child. So she must carry the mutant allele. And she's dead, so it may be either that whatever test, the diagnostic test couldn't be done on her, or she was non-penetrant, right? Yeah. Okay. Oh, how about this one? So we've got some telltale sign here. Signs, right? So we have 3 pedigrees.
Autosomal recessive.
Yeah, because
Consanguinity.
Yeah, so this pedigree, you can see, has multiple consanguinity there, yeah. So that'll get you an autosomal recessive disorder.
Two different.
Oh, and then here's a famous pedigree, the Royal Houses of Europe, hemophilia A. You can see all the affecteds are male. What's the inheritance pattern? X-linked recessive, yeah. Okay. All right, that's it. Thank you.
On some sunny Sunday afternoon, when you're reviewing these pedigrees, as you go through, I would make a list of the major modes of inheritance, and then make sure that not only can you figure out that mode of inheritance from the pedigree, but that you can draw a pedigree that reflects that mode of inheritance.
Under stress. Conditions of severe stress.
But that's not a sunny Sunday.
That's not a Sunday Sunday.
I also posted a chapter from the Human Molecular Genetics textbook on this stuff too, so please read that.
I can also post the PDF.